So I'm a pediatrician by training, and it's odd how I got into this research career. I moved to All India Institute of Medical Sciences from Lady Hardinge Medical College, and I met my mentor. And also, it's funny. I was taking a break because I had had a baby, and so it's how some incidents change your life. And I just joined as a research associate in one of the projects, and my mentor said that, do you want to do research? Do you want to do a PhD? And I said, no, I can't work with other people's children and use them for my research. And that was my beginning. And then I was told, it's not good to have your principles written on your forehead. They're never genuine that way. So that's how it began. And then, of course, this was in 1984, and I've come a long way. So what I'm going to take you through a story of how we tried to work for children and how we've been able to take some of our work into policy. And so before I start, I just want to say there were 4 or 5 things which shaped my career, which came from my mentor and came from the peer group that I was working with at All India Institute of Medical Sciences. That was essentially be an outlier, be unconventional in your thinking. You can be a physician, but you can be a physician researcher, which is not the usual thing we don't have too many in our country. Think of the larger picture. Think of what you can do for the country and the children and the larger picture. Always be uneasy about your success. Don't take it as the end point. You have to feel that, there may be something more that you can do. Always believe that you can do it unless proved otherwise. And don't judge by designations and positions, either people or yourself, because at the end of the day, you're only going to be remembered for the work you've done. And the last thing, which I think all of you know, that hard work will not kill you. So based on these 6-7 principles, we worked as a team. And I think in clinical research, it's the teamwork which really matters. But even there is some individual thinking and how you can coordinate and how you can take it forward. So just taking you back to 1985, when I walked into the pediatric ward as a team, there were disturbing images of children dying because of prolonged diarrhea. And when we moved into the community, we saw that these children were dying because of diarrhea more than 14 days. And so diarrhea is a continuum, but the deaths were happening when the diarrhea exceeded 14 days or more. So something was happening to the children after a certain point of time. And so we developed a conceptual framework and looked at various causes which could cause persistent diarrhea and a persistent mucosal injury. The thought behind it was, don't just get into that work. Think of what the concept could be and then try to solve each pathway as you go along. So we thought of, could it be just infections? Normally when you think of diarrhea, you would think of infection, and particularly persistent diarrhea, you'd say it's persistent infection. Could it be host factors? Could it be food intolerances and carbohydrate intolerances? Or could it just be a host nutrient deficiency? So our first thing was, is it infection? We walked into the community to look at the various causes of diarrhea, and we took weekly cultures of children with diarrhea, and we found that they had one infection, second infection, third infection, and then going on to have persistent diarrhea. And the funny thing was that children without diarrhea had similar kind of pathogens in their stool. The second thing was that a persistent single infection leading to persistent diarrhea was present in a very small proportion of children. The second thing was that small bowel bacterial overgrowth. It's said that developing country, prolonged diarrhea is because of our bacterial overgrowth. that time we didn't have sequencers and we didn't have microbiome being looked at. So just looking at culturable small bowel bacterial overgrowth, we found that those with persistent diarrhea and those without diarrhea had a similar kind of bacterial flora. We validated this further by evaluating the efficacy of large doses of oral broad-spectrum non-absorbable antimicrobials on small bowel bacterial overgrowth as compared to a placebo in randomized controlled trials in children with persistent diarrhea. And I'm not going into details, but just to show you that there was clear evidence that infections or small bowel bacterial overgrowth were not a major determinant for persistent diarrhea, and that antimicrobials had no role in its treatment. Now that was a major problem with the pediatricians and with the other physicians. How can you not treat persistent diarrhea with antimicrobials? So then we thought, could it be non-microbial risk factors for prolongation of acute diarrhea? Could it be dietary intolerances? So we did biopsies of children with persistent diarrhea. And I'll come to that in a minute, basically to see what are the enzymes there in the mucosa, and if they are decreasing, could that be causing intolerance? So we had these, and Satyajit sitting in the audience and any other physicians, I don't know, way back in 84 we didn't have endoscopes. So we used to have Crosby capsules, which were little capsules attached to a, you can see here, this is the Crosby capsule and this is a tube. So we had to make the patient, the baby, swallow that capsule and then make the baby walk around so that the capsule by peristalsis goes into the duodenum, crosses the DJ junction, reaches the jejunum, and then it had a very, I must say, innovative. You had to suck it, and with that sucking, there was a little blade which used to swirl on its own and take the biopsy piece with it. So it was a tedious process, and it took about 4 to 5 hours. The child had to be subjected to fluoroscopy, but we did manage to do a lot of those. And we found that the enterocyte height was decreased in those with persistent diarrhea, and the mucosal enzymes were less, and the lactase being more than the sucrase and glucomylase. So then we thought that if you're really seeing this in the mucosa, that means that all of them should be put off milk, because milk contains lactose, and that is what is causing the intolerance, and that is why they are having diarrhea. Because once you have undigested lactose in the gut, then it's going to be osmotic and lead to persistent diarrhea. But what you see in the lab is not necessarily what gets translated to actual actual life in children. What we found is that when we did, so what could we do? We could either give them a completely milk-free diet, or we could give them hydrolyzed lactose as yogurt, or we actually decrease the amount of lactose in the diet. The basis behind that was that the amount of lactose which can cause intolerance will depend on the amount of lactase per unit length of the intestine. So not all lactose is bad. Maybe small quantities of lactose could be given. But this could only be tested if you did it in the children. You can't really do it in the lab because the children have other factors and you have to take that into consideration. And so we tested this in randomized controlled trials, and we found that if you give diets with small quantity of lactose, which we called as milk cereal diet, small milk, large amount of cereals, because cereals help the digestion of lactose, we were able to develop a stepwise dietary algorithm which advocated simple milk cereal mixtures containing reduced concentrations of lactose as initial diets and leaving the more complex diets to the more severe children. So story has to have a conclusion. Were we to leave it there or were we to take it forward? And therefore, we then worked with the international groups and our government, and we were able to introduce this in the national government policy for treatment of diarrhea. And today, it's the standard teaching in textbooks that antimicrobials have no role in persistent diarrhea except for systemic infection. There's an evidence-based stepwise dietary algorithm. It's not consensus by the pediatricians. We had evidence to show that it should be stepwise. It's been tested further in many developing countries. It's been adapted for global use, and it's also part of textbooks and training. So the story continued. We had those who had persistent diarrhea, but we had those who had acute diarrhea who were going into prolonged diarrhea, and we had to see where that problem was and see if we could stop it. And dehydration is the biggest killer, and most of you know that the oral rehydration solution today is one of the biggest medical advances in medical research. And this came out of just an emergency based on cholera in the 1971 war. There hadn't been any evidence to show that the WHO ORS, which was earlier, I think you people would have been just born at that time, was being used, but it made a lot of difference to mortality. What we wanted to do was improve it, because the amount of sodium in the ORS that was present was causing hypernatremic dehydration for our children. And so we went on to look at different kinds of ORS, either fortified with amino acids or reducing the osmolarity of the solution by reducing sodium and glucose. This took us 10 years of work. And so one could be, there were some intestinal perfusion studies and experimental models. And again, what I'm trying to tell you, that it was lab to clinic at every stage. There were some perfusion studies in adult human volunteers, but what really needed us to bring it into practice and policy was evidence in adults and children. And so again, 10 years of work, we did clinical trials of oral rehydration solutions. The other question was, can we extrapolate data which has been generated in the West to our settings? We can't. We are a different population, we have a different gut, we have a different flora, we have a different disease. And we needed to get our own data. So we did a lot of these studies, and what I've shown you here is that we used to collect stool from our children, because the objective parameter for an efficacy of a product in diarrhea would be reducing stool weight. You can imagine a mother, the child passing 10 stools a day, and each stool about 20 to 30 ml, of even up to 50 to 60 ml, what kind of morbidity that's creating. And so we used to collect these stools and vomitus. We had these weighing machines. We were so paranoid about the amount of stool we were collecting that we were called the shitty gang, because in the ward of All India Institute, we set up this little oral rehydration unit and diarrhea unit. And here I just want to tell you I'll just stop and tell you that always believe that you can do it and believe in yourself. We were always told by our colleagues that we don't understand what you're doing. And I think it was only the confidence in ourselves, the confidence and some confidence in some people in us that yes, there is something that we're trying to do which would eventually go into practice and policy. And we had a mentor who was ready to push us till we die, till we had proved this. So we did this, and then finally the current oral rehydration solution that is in the market, which is being used by children for diarrhea and by adults for diarrhea, is the low osmolarity 245 milliosmoles per liter, which has now been used not only by the Indian government but by WHO as well. So this has been adopted in the practice and policy, and also being used in all developing countries for diarrhea. So again, the story didn't end here. Could we understand this whole syndrome better? And we started to wonder if the micronutrient deficiency, which was related, could that be related to increased incidence and severity of diarrhea? And we focused on zinc. That came from because many animal studies had shown that pathogen elimination is delayed because of zinc deficiency. We didn't do these studies. This was coming from other groups that were working on zinc. But what we found, that at least a third of children from our settings are zinc deficient. And there are many reasons for it. We have high number of stool losses. We have a diet which is high in cereals, so the absorption of zinc is delayed. Further, several animal and human studies had shown that zinc is critical for immune function and direct mucosal repair. Again, some evidence to show that it could be acting as an anti-secretory agent. So again, this evidence was coming from some biology and some work that had been done, but we needed to take it to practice, and that we could only do by again testing it in children. And we found that zinc given during an episode of diarrhea reduced duration and severity of illness. So here again was evidence, both from the lab and again in practice. And today what we have in the market and being used again by practice and in policy and in practice is the ORS. I'm sorry for using this trade name, but it's just all the ORS now. So we had to actually work with the industry. The industry was not ready to change a practice which had been there for so many years. And so they were using all kinds of ORS. But today, all the ORS available in the market is the ORS that I just showed you. The zinc was as a dispersible tablet. So we had to again work with the government, with Department of Biotechnology, transfer the technology of dispersible tablets. And today these tablets are being made in the country, and both zinc solution and zinc tablets are available and are being used globally. So the second story was that if you have zinc working in diarrhea, could it also work in other infections? After all, if it enhances immune responses, it may be working in other infections. And we went on to test zinc in treatment of young infant sepsis. And this was a large study in 700 young infants with clinical sepsis, where we found that zinc, if given as an adjunct treatment with antimicrobials, reduces the risk of treatment failure in infants with sepsis. We were fortunate that we could publish it in Lancet, and this is the story I was saying right in the beginning. We sent it to Lancet. It came back, revolving door. We said, we believe it's a good study. We believe we are good. It's very relevant for our country. At least get it reviewed. And the editors were very good. They got that paper reviewed and we were able to publish that. But again, as I said, don't end there. Your story has to have a conclusion, has to have an end. And now, so this is one study. It has to be reproduced. It has to be consistent. So, and also we looked at treatment failure rate in sepsis. The eventual thing is decreased mortality in sepsis. And so now we're evaluating in a large multi-country study efficacy of oral zinc in 4,000 young infants across India and Nepal to see how it can decrease mortality in young infant sepsis. Now, my question, what our group felt was that while we have enough clinical evidence, what we don't have is enough evidence to say how does it work in sepsis. Are there any, what are the mechanisms? And these were the questions that were coming up when we were working on that story. And at that point in time, we were put in touch with biologists, Dr. Satyajit Rath is here, Dr. Vinita Bal from the National Institute of Immunology. And we started to think that why can't we link this clinical with the mechanistic biology to understand the disease better, to be able to take it into, to be able to get your patients that benefit of the understanding that disease. And so there was this huge, as I say, as you must have seen, this valley of death between the clinicians and the biologists. And believe me, even at All India Institute of Medical Sciences, which is a multidisciplinary institute and one of the greatest institutes, I think, even though I'm not there, but there's always been this bridge. It was that the physicians wanted to approach the lab people if they wanted some assays done, and the biologists approached the physicians because they wanted samples, as we say, sampology. And that didn't work because the question had not been thought through together. We needed to actually evolve the research question and hypothesis together, with each team having a role. It can't be done in isolation. And That was the time when the Translational Health Science and Technology Institute was started, and I know it's not been presented in this in this meeting, but I just very briefly just to say that the vision is the institute started in 2010, so I've been one of the founder faculty, and was to study disease biology in humans and effective translation of alleles generated internally or by others, contribution to solutions that aid diagnosis, prediction, treatment, prognosis improvement, and what impacts public health. So my colleagues from NII, Dr. Rath and Dr. Bal, and some of my colleagues, Molly, and we were able to initiate this program on pediatric biology, which was an interdisciplinary program dedicated for maternal and child health to link mechanistic biology with classical epidemiology. I don't think there's any other formal program like this in the country other than this today. Through this, and the whole mandate is to build research on the biological basis of childhood health and disease so that it can create knowledge-driven interventions and technologies. And we've also been fortunate to develop through DBT glue grant programs where we've got links with hospitals, even though we don't have a hospital on the cluster at in Faridabad. So then was this whole story of if you have this platform, can you think of a large program which again will impact public health? I'll take another 5 minutes. So preterm birth is a serious problem in India. So is fetal growth restriction, and we, as you can see here, we are the first in global rank for number of preterm births, for deaths due to complications, and it also has long-term complications. So again, we need evidence which is unique to our settings that can be translated back. And for that, the question was, can we do it ourselves? So we developed a hypothesis that we need to stratify mothers for preterm birth for better understanding of mechanisms within these well-defined subcategories. Preterm birth is a huge syndrome, but can we identify subcategories in mothers where we can understand the biological mechanisms better to be able to develop preventive and therapeutic interventions. Also, I identify the modifiable clinical and epideterminants and then apply omics to understand important clinical and epi questions, understand the infection and inflammation of the disease. And incorporation of these biological correlates will help us to provide more reliable epi estimates and be able to help us to develop preventive and therapeutic strategies. Huge, ambitious program. I don't know whether I'm going to be able to have a logical conclusion by the time I retire, but it's been initiated. We've been able to build multidisciplinary teams for doing various parts of this program. We're following a pregnancy cohort of 8,000 women, and we're taking them up till birth and doing the various assays and that. But before that, I want to say is that why this idea was also important is that in the Pediatric Biology Center, we started to think that infection, as I said in the zinc sepsis study, that it's a major killer in the young infancy and childhood, and nobody really understands the immune system of a young infant. And we started this work in the Pediatric Biology Center to understand the maturity of the immune system. But that was from birth and onwards. What we added in this program is an antenatal period to the birth. So now what we have is an antenatal period and a birth cohort. So we understand the mechanisms that are happening before the baby is born and then follow it up to see how it affects the child. And we have, as you can see, multidisciplinary teams. We have the genomics component being done by the National Institute of Biomedical Genomics. We have the proteomics and the protein biochemistry and being done by the Regional Center for Biotechnology. of course, we are looking after the entire clinical program, the microbiome within THSTI and the microbial ecology. And the district hospital, Gurgaon, is the main study site, with the referral site being Safdarjung Hospital. So we've set up this entire research unit in the small district hospital. And what we've also done is set up a small research laboratory in the hospital for immediate processing of samples. Huge task, because you can imagine in a small district hospital, they don't understand research. For them, clinical care is the only thing that matters. So it has been a slow and steady process. We have set up an ultrasound imaging facility there so that we can do serial ultrasound images of these women. And this is not just for clinical care. The eventual aim is to use imaging as a biomarker to understand the disease of the antenatal period as well as birth and onwards. And this is the large clinical team. So when we talk about teams, we don't talk about 5 and 6 people. We talk about 50 and 60 people. And we have a large data management system now, because you can imagine the amount of data that will be generated, both clinical and biological. So we've got a data management system, which is where there's online data happening at the hospital, being transferred to THSTI and being saved here. And for this, we've set up a biorepository and an imaging data bank. So the program is 5 years, maybe more, with so many investigators. 8,000 women are going to be enrolled. 20,000 will be screened. We'll be doing something like 25,000 ultrasounds, about 32,000 contact visits. These are the lower limit, and about 1.5 lakh clinical samples will be collected. So this is the biorepository that we've initiated. These are the kind of samples that we're collecting, but this is again not stopping here. This is a platform where we will answer our questions through these samples, but this is a platform which will be used by other researchers for developing their own hypothesis-driven questions. Maybe outreach to other partners. I won't go into this. This is a diagnostic kit. This is another story which is very close to my heart, is on celiac disease. Very important childhood disease where children can't tolerate wheat. So you see these chronically malnourished children at the age of 10 and 12 coming at a height of 100 centimeters or 112 centimeters. And they cannot be diagnosed because the diagnostic tests are very expensive. So what we did was we collaborated with the biologists and created a diagnostic kit today, which is now available in the market for 1/3 of the price that is available. So in the market you get it for 2,000 rupees. To government hospitals, it will be provided at 60 rupees a kit. And this we feel has been a major contribution to child health. So just the last 2 messages. How can we do meaningful work? I think it's high risk and high reward. I mean, this is something that we've grown with over 30 years now. Move out of our comfort zone, accept big challenges. See, I'm coming from a very different field, so I can comfortably say this, that, big challenges for us is infant mortality, infant morbidity. And we believe that we can't read the path alone. We need to build bridges with different disciplines. My initial part, we did a lot of clinical work. My strength is that. But now, having moved to THSTI and sitting with other disciplines, my perspective has changed. And that has made a big difference to the way we are now shaping our programs. So, and lastly, I don't know how many of us are fond of diamonds. I am. And so are we going to be successful? We never know how close we are. So never give up your dreams. Thank you.